There are about 100 billion neurons in our brain and about 13.5 million neurons in our spinal cord. All of the neurons are each firing off 5 to 50 messages per second. This is all thanks to the electrical signal, the action potential, which allows our neurons to communicate with each other. In this presentation, we'll be taking a deeper dive into what a neuron is and how action potentials occur. And here are the sources we used for this presentation.
All right, so let's start off with what is a neuron? So neurons can be found throughout the entire human body. Many scientists classify neurons as information messengers, which transfer information throughout the body. This includes the information transfer of all seven senses. They transfer this information through electrical and chemical signals. The neuron is made up of seven different parts: the dendrites, soma or cell body, the nucleus, the axon, the myelin sheath, and lastly, the synaptic terminal or the synapses. Now let's move on to what is the resting membrane potential and how is it maintained. All neurons have what is called a resting membrane potential, which is about negative 70 millivolts. This means that the inside of a neuron is 70 millivolts less than the outside. This number can be attributed to the fact that there are relatively more cations, like sodium, outside the neuron and more anions, like chloride, inside. The resting membrane potential is maintained by two different channels, one being the sodium-potassium pump and the other being the sodium and potassium leak channels. The sodium-potassium pumps always pump sodium and potassium ions away from low concentration gradients into high concentration gradients. In order for these pumps to function, ATP is made from the mitochondria and used by the pumps. These pumps can be classified as active channels, meaning they require energy in order to function. On the other hand, the leaky channels are passive, meaning they require no energy to function and they happen naturally. This process can be defined as facilitated diffusion, which means that the molecules move from high to low concentrations. So these channels help keep the concentration/distribution of ions fairly constant, which is how the membrane potential is maintained.
What is an action potential and how does it occur? An action potential is the event the nerve cell uses to transmit information from one cell to another cell. In simple terms, an action potential is the increase of energy/electricity in the cell, which allows for neuronal communication. Once the neurotransmitter binds with the membrane protein at the synaptic dendrites at negative 70 millivolts, a voltage-gated sodium channel opens, which enables positively charged ions to flow across the membrane, raising the energy/electricity in the cell. Due to the increase in positively charged ions, the charge depolarizes inside the cell body, which subsequently causes the voltage of the neuron to increase. The charge across the membrane must reach the voltage threshold of negative 55 millivolts in order for an action potential to occur. If the signal is not strong enough, an action potential will not fire, as action potentials are all or nothing. They do not have degrees of magnitude. As the inside of the axon becomes electrically positive, the voltage-gated sodium channels become inactive and cannot be reopened until the resting potential or hypolarization is reached and ready to fire another action potential. This is called the absolute refractory period / repolarization. The voltage-gated potassium channels open, which allows the potassium ions to leave the axon, causing a decrease of charge in the membrane. This is known as the repolarization phase. It becomes more negative. At a certain point, too much potassium is leaving the axon, which causes the membrane to dip below the initial resting membrane potential. This phase is called hyperpolarization, in which the axon is in its relative refractory period. Subsequently, the sodium channels are ready to open again. However, in order to return back to the resting membrane potential, the sodium-potassium pump and potassium pumps use ATP to reset the membrane potential. Though action potentials can occur during hyperpolarization, they are more likely to occur when the neuron is in its resting potential because it is closer to its threshold. How does a neuron receive a signal and how is a neuron stimulated? As Max articulately stated earlier in the video, neurons are information messengers which transfer information throughout the body. But how do neurons communicate with each other? Well, once a neuron receives a prior signal, a chemical called a neurotransmitter is released to send the signal forward to the next neuron. Then, the neurotransmitter binds to a specific receptor on the front of the postsynaptic neuron, such as ligand-gated ion channels on the membrane of the neuron, allowing sodium and potassium to flow into the cell. Dendrites are branch-like structures at the beginning of the neuron which receive information and act as the antenna of the neuron. Based on the strength of the incoming signal that the dendrites have received, the neuron either lets in the signal or rejects the signal. Multiple dendrites receive information simultaneously. Therefore, if the combined effect of the signal received in all the dendrites changes the charge of a cell to reach thresholds, an action will occur. At the end of the neuron, there are synapses, which converts the energy initiated by the action potential into neurotransmitters.
Hi, my name is Quinn, and today I will be presenting on how action potential moves down the axon of a neuron. As the signal is received in the dendrites, it moves through the cell body into the axon hillock, and if the threshold is reached at negative 55 millivolts, action potential can officially occur, and the first sodium channel will open. The influx of sodium into the cell from this channel will trigger the next voltage-gated sodium channel to open. These positive ions will then cause the next passive ion channel to open, and so on and so forth. The reason that the signal can only move in one direction down the axon is because as the cell depolarizes, it is simultaneously in a process called inactivation, where the voltage-gated channels will become blocked by a polypeptide so that no sodium ions can flow into the cell, even though the channel isn't officially closed. This prevents the signal from traveling backward and keeps the signal strong enough to open the next sodium channel until it is time for repolarization and the sodium-gated channels are inactive and the signal is pushed by the electrochemical gradient into the synaptic terminal to be sent to the next neuron. The myelin sheath is made out of glial cells to insulate the axon to prevent the flow of ions into and out of the cell. The places on the axon that are unmyelinated are called nodes of Ranvier, and these are the only places along the axon where ions move between the neuron and the extracellular fluid. All of the leaky channels and sodium-potassium pumps are in these nodes, which all regenerate the action potential of the neuron. Saltatory conduction describes how the action potentials of an axon jump between the nodes and are accelerated by the myelin sheath. This is important for our understanding of the mind because action potential and this whole process are all really crucial in understanding how our brain functions and initiates command for the body to execute. How neurons communicate with one another and send signals is how the mind functions and is the most basic level of intelligence in all species. The human brain is capable of some truly incredible things, but all walks of life can experience hunger when they need energy, panic when they are in danger, and pain when they are wounded. The nervous system is the control center of the body and works to keep you alive and control your decision making. Understanding action potential is understanding what the signals and communication within these neurons actually is and how these signals are moved through the mind.